Well, it was a project that was launched in 1990 by the NTH, and that stands for National Institutes of Health. And the U.S. Department of Energy, to better understand all aspects related to the human genetic material, or DNA. In 2001, the sequence of the HGP was published. HGP is a scientific research project with the goal of determining the sequence of the chemical base pairs which makes up the human DNA and identifying and mapping all the genes of the human genome from a physical and functional standpoint. During the time that the HGP was formally introduced, there was an issue of the debate whether it would be more important to know the complete sequence of the genome or the known sequences should be better understood.
And to show you the true power of this molecule, we're going to start with one atom deep inside. Pull back and you'll see it form its A's and T's and C's and G's on the classic double spiral. And then starts the mysterious process that creates a healthy new baby. The interesting thing is that every human baby, every baby born is 99.9% identical in its genetic code to every other baby. So the tiniest differences in our genes will be hugely important and contribute to differences in height, physique, even talents, aptitudes, and can also explain what can break, what can make us sick. Cracking the code of those minuscule differences in DNA that influence health and illness is what the Human Genome Project is all about. Since 1990, scientists all over the world in university and government labs have been involved in a massive effort to read all 3 billion A's, T's, G's, and C's of human DNA. They predicted it would take at least 15 years. That was partly because in the early days of the project, a scientist could spend years, an entire career, trying to read just a handful of letters in the human genome. It took 10 years to find the one genetic mistake that causes cystic fibrosis. Another 10 years to find the gene for Huntington's disease. 15 years to find one of the genes that increase the risk for breast cancer. One letter at a time, painfully, slowly, frustratingly prone to mistakes and false leads. We asked Dr. Robert Waterston, a pioneer in mapping DNA, to show us the way it used to be done.
The original ladders for DNA sequence, we actually read by putting a little letter next to the band that we were calling and then writing those down on a piece of paper or into the computer after that. It's horrendous.
And we haven't mentioned the hardest part. This here, magnified 50,000 times, is an actual clump of DNA, chromosome 17. Now, if you look inside, you will find, of course, hundreds of millions of A's and C's and T's and G's, but it turns out that only about 1% of them are active and important. These are the genes that scientists are searching for. So somewhere in this dense chemical forest are genes involved in deafness, Alzheimer's, cancer, cataracts, but where? This is such a maze. Scientists need a map. But at the old pace, that would take close to forever. And then came the revolution. In the last 10 years, the entire process has been computerized. That cost hundreds of millions of dollars. But now, instead of decoding only a few hundred letters by hand in a day, together these machines can do 1,000 every second. And that has made all the difference.
This is something that's going to go in the textbooks. Everybody knows that. Everybody, when the Genome Project was being born, was consciously aware of their role in history.
Getting the letters out has been described as finding the blueprint of a human being, finding a manual for a human being, finding the code of a human being. What's your metaphor?
Oh, golly, geez. I mean, you can have very highfalutin metaphors for this kind of stuff. This is basically a parts list, right? Blueprints and all these things, it's just a parts list. It's a parts list with a lot of parts. If you take an airplane, a Boeing 777, I think it has like 100,000 parts. If I gave you a parts list for the Boeing 777, in one sense, you'd know a lot. You'd know 100,000 components that have got to be there, screws and wires and rudders and things like that. On the other hand, I bet you wouldn't know how to put it together, and I bet you wouldn't know why it flies. Well, we're in the same boat. We now have a parts list. That's what the Human Genome Project is about, is getting the parts list. If you want to understand the plane, you have to have the parts list, but that's not enough to understand why it flies. But of course, you'd be crazy not to start with the parts list.
And one reason it's so important to understand all those parts, to decode every letter of the genome, is because sometimes, out of 3 billion base pairs in our DNA, just one single letter can make a difference.
So there you see them talking about how important single letters can be, how far the Human Genome Project has come over the past 20 years, and hopefully what you took away from that was the intensity or the severity of the things that they're studying. They can now find genes that can be linked to cancer, cystic fibrosis, Parkinson's, Alzheimer's. All of these things can now be detected relatively early now, and I want to go over some of the methods to detect these diseases. The first is diagnostic testing. It's used to precisely detect what disease you have. So instead of looking at your symptoms and trying to compare your symptoms to various diseases, they can actually just look at your genome and straight up tell you exactly what disease that you have that's making you ill, and then from there they can plan your treatment or cure the disease that you have just because of the tests that are available. The second is predictive and pre-symptomatic genetic testing, and these genes are used to find a person's likelihood of developing a certain disease. So let's say your grandmother had Alzheimer's, and you want to know if you were going to run the risk of getting Alzheimer's. You can go and get your genome tested, and they can look at your genome and tell you the likelihood that you will develop Alzheimer's. If you will, if you won't, when you might be able to expect it. And it's not just limited to Alzheimer's, it's limited to almost every disease, thousands of diseases now. Another one is prenatal testing. So prenatal testing is offered during pregnancy, and it's kind of what you would think it is. They take the genome, the genes, the DNA from the fetus, and they examine the fetus, and it's essentially diagnostic testing and predictive testing. They'll tell you what if any problems your baby's going to have. Is he autistic? Does he have Down syndrome? Does he have Huntington's disease? Is he going to have cystic fibrosis? They can look at all these things and tell you if your baby is going to be healthy when born, and if he's going to be healthy later in life, which is, to me, is amazing because it's just such a large window that they can look into from a baby. The next is newborn screening. It's very similar to prenatal testing, except the baby's already born. It's the same thing, it's just diagnostic testing. And predictive testing, and they're just looking to see, is the baby healthy? What genetic diseases does the baby have, if any? What genetic diseases is it likely to get? And then pharmacogenomic testing. It gives you information about how certain medicines will affect your DNA structure. So let's say you're the kind of person who has side effects. With every medicine you take, you always get negative side effects, or you get allergic reactions to a lot of the medicines that you take. With this kind of testing, they can test medicines with your genetic structure and figure out what medicines are not going to harm you, what medicines are going to benefit you, and from there, your healthcare provider can start helping you decide what prescriptions you need to be taking, what medicine you need to be taking that's best going to suit you, instead of just a trial and error method. And if you've ever been allergic to a medicine, a trial and error method is not fun. It's actually pretty scary. And then the second to last is research genetic testing. And it's more used to figure out what contributes to your health or a disease that you have. So they can tell you what genes that you have that can be contributing to you being overweight, or what genes can be contributing to your ability to put on a lot of muscle or have good muscle mass. It's a lot less precise, but it's the information I still think is regardless. And then that brings me to what I believe is the most important, and that's carrier testing. Carrier testing is used obviously to test who carries a genetic disease. It does not necessarily mean that you have to have that genetic disease to carry it. So I want to give an example. In my psychology class last semester, we were learning about a woman, and when her mother was born around the age of 10, she got very aggressive ovarian cancer. At the age of 10, almost killed her. But she lived a healthy life after that. Got married, had her daughter, which is the woman I'm now speaking of. When that woman was 10, she had the exact same form of ovarian cancer around the exact same age, and almost killed her too. But she lived a healthy, or she overcame it, lived a healthy life. Because of the Human Genome Project, her and her partner were able to go to a facility where you can get this testing done. They got mouth swabs done, and scientists were able to combine her genome and his genome, look at all the possible genetic outcomes for their children, and they were able to determine that should she have a daughter, there's a 75% chance that the daughter will have the exact same form of cancer. So with this information, they then in vitro fertilized eggs, took a DNA sample from the fertilized eggs, and were able to get her a daughter that did not have or carry the cancer gene that her and her mother had both suffered from. Now the benefits of that, as you can tell, are colossal. I mean, knowing what diseases that you have isn't always a comfortable thing to hear. Or what diseases you might get. Not everyone wants to know if they're going to have Alzheimer's. Not everyone wants to know if they're going to get cystic fibrosis or their likelihood to get breast cancer or any kind of cancer. Some of it can be disheartening. But I feel like the benefits outweigh the pros in so much. Knowing what you're going to have, I think it provides peace of mind for a lot of parents, knowing that they can have healthy children without having to worry about them even in the later stages of their life. And then the negatives. I think this is where the most controversy comes from with this issue. Look at someone like Stephen Hawking. Had the Human Genome Project been done 30 years ago and been prominent in our society, people like Stephen Hawking wouldn't be alive, because they would have seen that he would, I'm not 100% sure on his condition, but they would have, I know it's an identifiable condition, and he wouldn't have been born. There's people with dwarfism that wouldn't be born that are alive today, people with cystic fibrosis, Alzheimer's. Those people would not have been born if this was around a long time ago, but they are born, and I think looking forward, this is a vital tool in determining the health of children, which is going to take us to Laura.
So what does this mean for babies? For future generations, I think that genetic engineering is going to be a big concept that people are going to be playing around with because it's really cool. You can choose the traits and the genes that your children get to determine whether or not they'll be healthy or carry certain diseases. So there's two different ways to alter the genetic makeup. There's somatic, which roots from the word soma, which is a Greek word that means body, and it affects specific genes and specific organs and tissues that only affect the person that would be, it would only affect the embryo that's being genetically modified. It wouldn't affect their offspring or anything. So any diseases that would be prevented for that person could show back up in their children's genetic makeup. The second way is called Germline, and this targets the genes within the egg and the sperm, or even early on embryos. And this modifies your genes so that it will continue to affect your offspring as well, and not just yours. After a baby is born, they do screenings for them, and this can test for 56 different types of diseases, but not all states test for all diseases because of the costs. Some states only test up to 30 different kinds of diseases, but the diseases that they test for are only treatable, they aren't curable. So even if you were to find out that your child has a disease, some people like to argue that it wouldn't be very beneficial. But the screenings are done 1 to 2 days after a baby is delivered, and they get blood drawn from their heel to do the testing for things such as cystic fibrosis or muscular dystrophy, even sickle cell disease or hypothyroidism. But those are just a few.
Is China genetically engineering their babies to be smarter? Who isn't? Hey friends, Trace here for D News. The internet is abuzz with talk about genetically engineered babies, thanks to a piece in Vice by Alex Error. China is planning to genetically cultivate their population. The internet seems to think that this article is proof the Chinese will inevitably genetically modify their population into super beings. And as we know, the internet is never wrong. If we select to keep people from having major genetic disorders, it's okay, but not if we pick for height or beauty or athleticism or brain power. The Vice piece revealed that a company called BGI is looking into the human genome to find the genes that control intelligence. They'll then be able to select the most intelligent sperm and egg from a couple and use that for their baby. It's not actually genetic engineering. They're not playing with the DNA. They're just spotting the best DNA and purposefully selecting it. It's very much like how farmers genetically modify their cow herds by breeding the best milkers or beefers or whatever together. Stephen Su gave a talk at Michigan State University about this topic. Definitely check that out. He explained how very soon we will all have enough genetic knowledge to do this with our children. The cost of gene sequencing is getting exponentially smaller, while the power of computers is doubling. In the not too distant future, maybe the next 10 years, we will be able to sequence a whole genome of sperm and egg and look for specific genetic markers. Sue shows that even with coaching and tutoring, there's not a huge statistical difference in test scores. But if you could select to impregnate with the best sperm for intelligence, you might bump it up a bit. And after several generations, you could conceivably up the smarts of a whole population. It's also important to note that China still maintains a policy where, unless you pay a fine, you're only allowed to have one pregnancy. So in the minds of new parents, they just want the best kid that they can get. From what I've read, selecting for desirable traits isn't unusual. Ultrasounds are routinely done both here and in China to learn how a fetus is developing. And if developmental problems are discovered, some choose to end the pregnancy. What Sue and Vice are discussing is the idea of filtering even earlier in the process. The scary thing is the Gattaca idea, where humans are selected for specific traits like intelligence, and those who choose not to do that are deemed less human. My concern is the natural way things are done might not be perfect, but until we know more about why a sperm picks an egg and how that process happens, we might select for things that are good for society, but bad for humanity and our species. Would you want to pick a prodigy for your progeny? Let us know in the comments, and be sure to check us out.
So if we are able to choose the genes and traits that our children have, who's to say that we can't create like super babies who are super strong or super intelligent? But that leads to problems like what's going to happen when those babies have babies of their own? Will they have genetic diseases? We don't know, we aren't sure yet. And I think that could lead a lot of people to fear eugenics or like deliberately attempting to improve the human race through artificial genetic selection or selective breeding. And although the idea of eugenics calls to mind the horrors of the Nazi regime, both sides of this debate have legitimate arguments highlighting the pros and cons of attempting to create a strong, healthier race of human beings.
In conclusion, scientists are still finding fundamental surprises, and despite the unknowns, and researchers are beginning to use genome data to unravel one of medicine's greatest mysteries. The gap between the promise of the Human Genome Project and the realization of that promise, that will narrow as researchers will disdain the complex and exquisite details of the genetic genome landscape and the conditions that shape it. That's all.